Okay, great. Welcome everyone to a very special issue of the Journal Club. We have a guest speaker today, Prof Theresa Rossouw at University of Pretoria. They sent out an advert with a very long and impressive list of qualifications, but very briefly, Prof Rossouw has a medical background. She works as a clinician. She has 2 master's degrees in the areas of philosophy and in public health, 2 PhDs in philosophy, one at Stellenbosch University and a PhD in immunology at UP. So she has a diverse area of interests, but she is at the forefront of HIV research and consultant or clinician with patients at Tshwane area in Pretoria. And just recently with COVID-19, she's also been on the battlefields and the front lines in that fight. So she has had her hands full being a clinician and a researcher at the same time. And there are various societies and associations she's part of. She's also is part of the South African Research or Research Ethics Committee, co-chairperson, in fact. So, yeah, NRF rated scientists awarded a silver medal by the South African Medical Research Council in 2023. So, in that list that we sent in the advertisement, just go through it and you'll see how impressive her resume is. And I know Prof Rossouw has another engagement later, so we're going to jump straight into it and hear from her what she has been doing lately in her research lab. So please, Prof Rossouw, share with us.
Thank you so much, Shane. Thank you for the lovely introduction and for the opportunity to speak to members of this journal club. I really value it very highly. For some reason, I'm not managing to switch my camera on. Maybe towards the end, I'll get that going. But can I just check that you can hear me and you can see the slides all right?
Yes, all is good.
Okay, fantastic. So what I'm going to try and do is just give a very broad overview of what we've been doing in the HIV immunopathology laboratory. I'm very fortunate that I work with a wonderful team of people. No man or woman is an island, so it's really important to be in an environment where you can flourish. So as Shane said, I'm a clinician scientist, so my big love is really treating patients. But for the last 8 years or so, I've spent more and more time in the laboratory, and it's also given me a very good understanding of, how the two should work together, that one cannot ignore the one, it will be at the peril of the other. So I've been working on this puzzle of HIV for a long time. So I started off by being the principal head of an HIV treatment clinic here in Tshwane. It was part of the academic hospital. And I learned so much about, just the diversity of disease that I started putting together the pieces for myself of what I think would be critical to understand for us to really get to grips with this virus. So all I'm going to talk about will really be focused on HIV, and I'm going to have these 4 puzzle pieces. I'm not going to talk about ethics because I know that's not really that relevant, but that is also one of my passions. So I will be talking about systemic immune activation, co-infections, and then also drug resistance, all in the context of HIV. So the pieces of the puzzle for me look like this. Under immune activation, we've done quite a lot of work on microbial translocation. We're also increasingly interested in what happens on the platelet endothelium level. And we've done quite a big study on children that are exposed to HIV but uninfected. So those are the children, we call them HEUs, so HIV-exposed uninfected babies, and why their immune system is actually not functioning the way that it should. And then also doing some work on HIV viral reservoir. In terms of infections, I've done some work on tuberculosis and hepatitis B, but I really won't call myself an expert there. For the last few years, we've really been focusing on SARS-CoV-2 and how that interacts with HIV. And then drug resistance, it's fortunately less of a problem at the moment because of dolutegravir, but it is sure to come back again. So we've done a number of studies on transmitted and acquired drug resistance, both in adults and children. And I will tell you a little bit about the studies that we are busy with and that are starting up at the moment. So if we first just look at the first piece of the puzzle, I just briefly want to explain what immune activation is and why I think it is important. So if we look at this graphic, this shows acute HIV infection on the left. And just to take you through it, so the blue line would be the mucosal CD4 cells. We see that they are depleted very early in infection, and they actually never recover. Once they have that initial insult, they continue to cause problems for the immune system lifelong. In the red, we have the HV viral load. So after initial infection, the body is able to control HIV for quite some time. And then gradually over years, the viral load starts picking up as the CD4 cells are depleted. In purple, you'll see the blood CD4 cells. So there's an initial insult, but not nearly as badly as in the mucosa. And for all practical purposes, these cells are fine for up to 5 to 10 years into infection. And it's only when they start dropping down that people start getting significant clinical disease. But what I want you to focus on is that black line of immune activation. So this immune activation happens together with acute infection, and that never settles. It stays at extremely high levels. And even on antiretroviral therapy that is virally suppressive, we still find that immune activation persists. And this is the major driver of HIV-associated mortality in terms of cardiovascular disease, renal disease, liver disease, and so forth. Now, when we think about the pathogenesis of HIV, we know that it is the host and not really the virus that determines whether disease progresses. And we know this from people that are slow progressors or fast progressors. A lot of it has to do with how the body responds to the viral infection. It's the same infection, but people do differently. So if we just look at the chronic immune activation, the animal models, so in the non-pathogenic form, so these are animals that are infected with SIV, the simian immunodeficiency virus, but they don't get ill from it. So they have high viremia, their CD4 cells are depleted, but they don't have immune activation. And for that reason, their CD4 counts remain normal, and they do not develop acquired immunodeficiency syndrome. If we compare that, on the other hand, with the pathogenic form, so these would be rhesus macaques, also infected with SIV, they have high viremia, they get the same depletion of the CD4 cells in the gut, but they also get immune activation. And because of that, we believe they have a decline in their peripheral CD4 counts and they develop AIDS. So we think that the mechanisms that are driving this immune activation might actually hold the key to understanding the HIV pathogenesis. So the areas that we've really looked at in terms of systemic immune activation would be these four, and I'm going to show you each of them, the studies that we have done and the ones that we are currently busy with. Unfortunately, I won't have time to go into any of the results, but in next Journal Club sessions, some of my students will be presenting. And then we can really delve deeper into the results of these studies. I'm just going to try and give a very global overview. So if we first look at microbial translocation, this is a very busy slide, but all I want you to focus on is that we have the gut inflammation because of that insult of the mucosal CD4s. And because of that inflammation in the gut, we have microbial translocation. So what that means is that there are not whole bacteria usually, but portions of microbial products that get into the systemic circulation where they do not belong, and they continuously trigger inflammation. Together with the inflammation, they also trigger coagulation. So you often have immunothrombosis, and for that reason, people get endothelial dysfunction and inflammation, and that often leads to cardiovascular disease. Now, if you combine this with other factors such as smoking, obesity, substance use, co-infection with other pathogens such as hepatitis C virus, then this accelerates this whole process of endothelial dysfunction and inflammation. So this leaky gut idea was originally shown to be a real problem in American patients. We were the first group to show it in an African population. And we actually postulated that it would be more significant here because we have more enteric pathogens and also helminths that may be skewing the gut immunity as well. So on the one side, we have this epithelial barrier disruption. On the other side, we have the loss of the mucosal immunity. And we know that the T helper 17 response is severely affected. And this causes this continual translocation of these microbial products. So if we look what happens on treatment, so on the left-hand side in blue, those would be people who are uninfected with HIV. In the middle in green would be people that are living with HIV and that are on ART. And then on the final side in yellow, you'll see people are not on ART. And what this shows is that even though the microbial translocation decreases when somebody is on ART, it never corrects, it never goes back to normal. And the way that we measure this, there are many ways, but this was done, for instance, with lipopolysaccharide, quite a complicated test to do, but a useful test. We've moved away from LPS a little bit just because you need to collect the blood in LPS-free tubes, which can be a problem if you are depending on the clinical services to provide you with blood. So we have moved towards other markers like LPS binding protein, as well as intestinal fatty acid binding protein as well. So we've published a number of articles, and as I said, this was quite a while ago. This was 2013 already, where we showed this for the first time in an African population. We did gut biopsies looking at physical markers of gut integrity, and then also looked at the immunology thereof. So just very briefly, I said I'm not going to go into detail, but just to show that there is persistent immune activation. Even after 1 year of ART, we still had elevated levels of tumor necrosis factor, which is highly pro-inflammatory, but also soluble CD14, which is a monocyte activation marker that we think is driven by microbial translocation. We've also looked at whether we can find a signature. Is there maybe an immune signature that one can test for to distinguish people who are suppressed versus those who are non-suppressed? And we did come up with a 5 biomarker signature. But, to be honest, it doesn't have a lot of value above doing a viral load test in terms of cost. So that is not something that we took much further than that. We've also looked at children and in their mothers. And in this population, we similarly found that there were elevated levels of a number of cytokines that we've looked at, principally being soluble CD14, once again, beta-2 microglobulin, CRP, IP10, TGF-beta1. So a lot of cytokines that I'm not going to go into detail now, but just to show that there were differences in the mothers when we compared them to adult controls without HIV. And then there were also differences between children and pediatric controls, where we found significant differences in the majority of these cytokines that we assessed. Once again, also showing here that some of these markers never settle, they never go back to a or on treatment, they never go back to normal levels, so to speak. They stay elevated. For instance, the anti-inflammatory marker TGF-beta1, and once again, our big culprit, the soluble CD14. Now, you may also know that this is linked with cardiovascular disease. So it's one of the markers that we can use together with CRP and IL-6 to look for an increased risk of cardiovascular disease over time. So the second aspect that we looked at was, what does all this inflammation do to platelets and the endothelium? Because we're dealing with cardiovascular disease, we know that it is critical that the endothelium should be healthy. Once again, I'm sorry, this is a bit of a busy slide, but just to show you, this was created by one of our students in BioRender, so thank you, Crystal. So just showing that the innate immune system releases a lot of molecules chemokines, growth factors, and cytokines that can activate platelets. And in our work, we found that two specifically remain elevated in people living with HIV, and that's RANTES and soluble CD40 ligand. And also, if we look at the adaptive immune system, there are also cytokines that are elevated and then can increase the risk for cardiovascular disease. We are specifically interested in the new medication on the market, so dolutegravir. Dolutegravir is an integrase inhibitor, and it is being used extensively. It's now being used for children and adults in first and second line. So we will be seeing a lot of this drug if we think that about 8 million people in South Africa alone are living with HIV. And the reason we worry about this drug is a PhD student of mine, Maurice Madzime, did work in the laboratory, so purely in vitro work, where we looked at the actions of various antiretroviral agents on platelets and neutrophils and markers of endothelial activation. And in all of the medication that we tested, all the ARVs, dolutegravir really stood out by potentiating platelet activation. And we are postulating that it could be through a calcium-dependent ionophore-like mechanism. And this is, of course, really problematic for people who might already have an increased risk for cardiovascular disease. If we further potentiate this platelet activation, this could, of course, cause significant problems. We also found some differences in neutrophils. Once again, the same kind of mechanism where they are highly activated in at least cells that we treat with dolutegravir. We have not shown this in people yet. So, oops, doesn't want to advance. There we go. So at the moment, we are busy constructing a study together with one of my previous PhD students, Sanele Ngobo, who finished his PhD last year. He's a clinical associate, and together with him and another PhD student that we are co-supervising, Edward Labello, we are planning to set up a prospective cohort of people living with HIV that are newly initiated on dolutegravir, and we want to follow them for 12 months and look at markers of cardiovascular risk together with traditional risk factors, and then look for markers of platelet and endothelial activation. One of the slides I wanted to include here is there was a recent article, recent being last year, of 1.68 times increased risk of cardiovascular disease in people with no other underlying risk factors once they transition over to dolutegravir. So that was really the reason that we wanted to investigate this drug. So we're in the planning stages, and hopefully we will be able to start recruiting early next year. Now, a big focus of ours has been on children who have been exposed to HIV and antiretroviral therapy in the womb. So we feel this is really critical because if we just look at the statistics, about 5 million women are living with HIV in South Africa, and about 29% of them are living with HIV. And if we look at this, the number of births per year, this means that almost 350,000 children are yearly exposed to HIV and antiretroviral therapy. We used to think that this was not a problem and that these kids, we discharged them home because, you know, they were now uninfected and that was the ultimate prize. But a colleague of mine did her PhD on the growth of these, of children living with HIV, using this exposed but uninfected group as a control. And then they found that their growth was actually significantly impacted. So we've further seen that they have increased morbidity and mortality, especially of infectious diseases. And they often get the same kind of infections that a child would get who is living with HIV, which is quite strange. They often also tend to have neurodevelopmental problems and suboptimal growth. I spoke a little bit about the immune dysfunction, but it really is in both the adaptive and the innate immune system. And we're trying to tease those apart. They have high levels of inflammation, and then they also have some metabolic abnormalities. So we only recently started delving into the metabolic side of things together with Shane and collaborator, Ruan. And we've really been wonderful in helping us to try and understand this very complex biology. So to understand this problem, we developed quite a large cohort. It doesn't sound that large, but I have to tell you, it's incredibly difficult to get all of this together and quite expensive to do. So we developed a cohort that we call Siakula, which means to grow or to flourish. It's a prospective cohort study. It consists of a multitude of disciplines, obstetrics, pediatrics, ourselves, family medicine, nutrition, food sciences, statistics, and so forth. We recruited 305 mother-infant dyads, and we just completed our second-year follow-up. And we're trying to tease out, if you can look on the right, you know, all the multiple confounders that we could find in these children. So firstly, they're exposed to HIV, then to antiretroviral therapy. Then there are maternal factors that could influence how they develop, for instance, maternal age, maternal nutrition, the lifestyle of the mother. And then, of course, postpartum, there are also factors such as the quality and duration of the quality of breast milk, the duration of breastfeeding. And then once again, other confounders that would be lifestyle and socioeconomic mostly. So together with the SAMRC Research Center for Maternal, Fetal, Newborn and Child Health at the University of Pretoria, we put this together, and this is a very basic overview of how we're trying to see, is it microbial translocation? Where does that fit in with the chronic inflammation? Is there maybe impaired T cell function or B cell function? And how does all of this work together to lead to poor fetal growth and low birth weights, and sometimes also preterm delivery? So there are a few factors that we are considering. The first is, of course, the HIV exposure and the exposure to antiretroviral therapy, but then also exposure to other pathogens and other persistent viral infections, such as CMV and Epstein-Barr virus, for instance, that we know could play a role. So we sampled mothers and infants quite frequently. This just gives a summary. At 28 and 36 weeks, we took blood samples from mums. We also took vaginal swabs, and from the blood samples, we took off PBMC, peripheral blood mononuclear cells, PBMCs, and we stored plasma, and we also have serum. And then at birth, we did the same, but we also get colostrum, so the first milk, and we have the placenta. And then we take cord blood that we look at various immunological markers as well. And at that time point, we start doing whole blood stimulation testing, because we want to look at the function of the cells. Are they able to respond to pathogens? So we want to see, are they there? What do they look like? But then also, how do they respond to a pathogenic challenge? And then the mums and the infants we follow very frequently. We sample again at 6, 10, and 14 weeks, and then at 6, 9, and 12 months, and finally at 2 years. And we try to collect stool samples when that is available. We also get breast milk samples, and we're very interested in looking at the microbiome in all of those samples as well. This is slightly outdated. I'm not quite sure where we are now, but just to show you that we have collected a vast number of samples and stored them under very good conditions at -80 or in liquid nitrogen dewars. So we have more than 5,000 plasma samples. So that's all the aliquots that we've made. Also PBMCs and serum. We've done 379 whole blood stimulation tests, more than 1,000 vaginal swabs, and we have vaginal swab slides as well. A smaller cohort of stool samples that has proved to be very difficult to collect, but we do have a cohort that we think is usable, and then more than 600 breast milk samples. So now the big job is, of course, to analyze all of this and to make sense of it. So we've been doing flow cytometry, and we have quite a comprehensive T cell panel where you can look at T cell phenotypes, but also specifically at their maturation. We have monocytes that we also look at, and in this panel we can also look into the classification and also the activation of these cells. And then we do cytokine and chemokine analysis with a Luminex panel at various time points. So various student projects are linked to this study. We have at least 3 PhDs. I'm not giving the clinical PhDs that are part of this as well, just the ones that have a laboratory component. So one is Louise, that's working together with Ruan and Shane, doing the metabolomics of some of these mothers and their infants. Felicia is an obstetrician. She's doing the antepartum growth. So we have antenatal sonars that we look at the growth of the liver, the thymus, and then, of course, just regular anthropometry from 22 weeks gestation. And then Andrea is doing the flow cytometry, and we're looking at markers of postnatal growth and seeing how that correlates with the T cell and the monocyte phenotypes. We also had a number of master's students on this project, 3 of the 4 that have completed now. So Brandon did work on the whole blood stimulation testing, Shanae did breast milk analysis of the cytokines, Ivan did flow cytometry, and Namonde is busy with postpartum growth and looking at monocyte polarization profiles. And then finally, on the immune activation, we also have a study where we are working with colleagues in the US looking at the reservoir. So it's called the Mechanisms Controlling the Persistence of Infectious HIV Reservoirs in Children. It has a very interesting hypothesis, I think. So this was all my colleague in the US, Lisa Frankel, who it was her brainchild. So there is a concept called maternal microchimerism. And this is where maternal cells persist in the infant. So we know that during pregnancy, there is a mixture of cells. So fetal cells end up in the mum and maternal cells end up in the fetus. And a proportion of these cells persist lifelong in the offspring of a parent, so of a mother specifically. So the theory is that if HIV comes in through one of those maternal cells into the infant, the body will automatically protect that maternal cell. It will make it into a sanctuary site because it doesn't want to attack that cell. And that will possibly allow for HIV to have a very safe reservoir where it can then resurface from when it is ready. So that is the main hypothesis. How we fit in is we are looking at the imbalance between the T regulatory and the T helper 17 profiles that alternatively drive tolerance and inflammation. So we're trying to see, is there a more tolerant environment in the women where we do find maternal microchimerism compared to those where we do not find the maternal microchimerism in the infant? So as I said, this is in collaboration with Lisa Frankel from Seattle Children's Hospital. We recruited 49 mother-child pairs and 30 healthy control for the children, and they were aged matched. We followed them at the first visit and then 3 monthly for 3 years while they were on treatment. So they had to be virologically suppressed on treatment. And we want to get as many cells as we can because we're looking for those rare chimeric cells. And that's why the follow-up was so extensive. So we've collected blood. Once again, we stored PBMCs and plasma. In the US, they're doing the detection of the microchimeric cells. They're also doing integration site testing. So this is the other hypothesis behind it. So depending on where HIV integrates into the genome, if it integrates into a very tolerogenic area that will probably suppress inflammation, then those cells will be more likely to persist as well. And then we are doing the translocation marker testing. We're looking for microbial translocation and then the cytokine analysis. Sure, so that is the first one. That is the immune activation, and that has really been the bulk of my work in the last 10 years. If we then very briefly look at co-infections, I'm just going to focus on SARS-CoV-2. We have a number of cohorts that we recruited during the pandemic. We're working together with Veronica Ikeman, who's the head of the ICU and the infectious diseases unit at Steve Biko Academic Hospital, as well as Farid Abdullah, who's from the MRC and also doing sessions at the ID clinic. So we have an adult cohort that we started recruiting in May 2020. And we recruited a total of 214 participants, 43 of whom were living with HIV. We collected blood on the day of admission and then also 7 days after admission. And then we stored plasma, buffy coat PBMCs, and we also did dry blood spots. And we've been doing flow cytometry and cytokine analysis and finding very intriguing results. We're actually very interested in seeing whether people living with HIV actually do worse than those who do not have HIV. But we can't find a difference. It actually seems to be the opposite, that people who are well controlled on their treatment actually do a little bit better. And we're hoping to publish that article. I just got the reviews back this morning, so we have a little bit of work to do, but then hopefully that will be published. Then we also recruited a pediatric cohort. They present quite differently to adults. So we have 78 patients in that cohort. You'll see quite a distribution in terms of age. And we collected 2 EDTAs, once again did the same kind of storage, and also busy with flow cytometry and cytokine analysis on those. So the adult cohort is forming the PhD of Mieke van der Meest. And then the pediatric cohort is Janae Klutter, who's a pediatrician who's obtaining her PhD in clinical immunology. Then we're also looking at patients with long COVID. Now, this is work in the making. And we didn't plan to do this, but we've had so many colleagues that have been severely affected by long COVID. And we've been asked to, if they're not something that we can do to look at their immunology, to try and explain some of these symptoms. So to date, we have 12 patients with quite an age range. And once again, the most fascinating results. We are seeing a hyperinflamed and exhausted immune system in these patients with high levels of cytokines, even in some instances higher than in those with acute COVID. So something is clearly keeping this immune system activated for some reason. And to try and tease that out, we're working together with PhD Pretorius at Stellenbosch University. She's been doing a lot of the blood clotting. So she was the first person to publish on the presence of these microclots that these patients have. Now, the tricky thing with them is no test comes back abnormally. So our regular clotting tests will all be normal. And you need a specific fluorescence microscopy to look for these microclots. We're also working with Tulani Malakanyane here at the University of Pretoria into the microbiome. He's the Saatchi Chair in microbiome work. And we do postulate that these patients have this, as we saw with HIV, that they have impairment of their gut integrity and continuous microbial translocation. And for those of you who've been following the literature, you might have seen that the gut is also postulated to be a reservoir for SARS-CoV-2, that in some way it can persist in the gut. And then that could be one of the reasons that people have persistent symptoms. And then, of course, we have to look at the metabolome and how, just going deeper and deeper into the pathology. And for that, we are working together with Ruan and Shane to try and understand the metabolomics behind it. We have a COVID-19 long-term follow-up study, and we are planning to recruit patients for the long COVID through here. So these are people that were admitted, and then we followed them prospectively over time. You'll see multiple time points up to 12 months. And we have samples, and our plan is now to go back to them and to understand whether any of them qualify or meet the criteria for long COVID. And then we can include them into our other cohort as well. And we've been most fortunate to work with the NICD on this, so specifically Penny Moore and her group at the NICD have been absolutely fabulous looking at the antibodies and also looking at differences in people living with HIV and without HIV in terms of antibody responses. We just embarked on a study this year where we are very concerned about people living with HIV, but also those with other immune deficiencies, such as people who are on chemotherapy, those who have autoimmune disease, who may be on disease-modifying therapy, or those that have renal failure and that may be on immunosuppressive therapy because of a transplant, but also on dialysis. So we're trying to understand what happens to them when they receive a vaccination for COVID-19. We're looking both at J&J and also at Pfizer. And we are doing, so people are encouraged to have their booster, as they should in terms of the national guidelines. And then we take blood pre-boost, and then after 6 weeks and after 6 months. We are looking at the T-cell function on this side, so we will be doing T-cell stimulation testing. And there on the left is Kanisile Chabalala, who's the PI of the study. And once again, Farid is instrumental there. And Penny will be doing the B-cell work. And then very briefly on drug resistance, I'm not going to talk about any of the previous work. I just want to show you what we are busy with at the moment. So we have one study that's being led by Louise Dutoy in our lab, where we're looking at dolutegravir resistance, emergence, and adherence monitoring. It's once again in collaboration with Lisa Frenkel at Seattle's Children's Hospital. We are planning to recruit 150 participants, and we follow them prospectively. We're looking for people who are not suppressed on dolutegravir. So the common theory at the moment is that people will not develop resistance to dolutegravir. You know, as clinicians, we're always very optimistic about these new drugs. We think it's going to solve all our problems. It's going to be the best thing ever. But we know, of course, that resistance will develop. But the pathways are a little bit unclear still. So we do postulate that these patients will develop resistance in areas outside of the target gene, which is integrase. So we are looking at other parts of the genome to see if we can find drug resistance mutations of relevance. So that is the one study. And then the final one that we just submitted a grant for, and I'm holding thumbs we will get, is point of care treatment monitoring, where we are looking at urine levels of tenofovir as a point of care test. And then introducing, that has already been shown to improve adherence. But we want to compare that with a dolutegravir level dry blood spot test and to see which one comes out better and whether we can actually use that as a way to improve adherence. Because if people can get the results directly when they are at the clinic, we can tell them, look, your blood levels are not high enough, you need to take your medication better. We believe that that could lead to a very nice and hopefully affordable tool that we can use to improve adherence. Great, and that is in a nutshell from me, just an overview of what we've been busy with for the last 10 or 15 years, and I'll be happy to answer questions.
Thank you so much, Prof Rossouw.
I found my video. Sorry about that.
So is that all you're doing? I mean, that's like, you know, just an afternoon's worth of work. There's so much work you are doing, and still a clinician at the same time. It's unbelievable. So yeah, Prof Rossouw has another engagement, so she can only stick around for another 5-6 minutes. So anyone who has any questions, you can raise your hand and you can ask her directly. Her email is on there. So I'm sure she would appreciate anyone communicating with her, questions on what she's doing, possible collaborations, any potential students out there who want to join in with Theresa's research. You see, transdisciplinary is even an underused word here in her line of work. So she is across all the disciplines, and it's quite amazing what she is doing. And she mentioned next week, one of her students is going to give more of an insightful talk on how they've been using metabolomics in their research. So that is next week. And Prof Rossouw just gave an overview of everything she is doing and done. So I encourage people, I know she's a very intimidating woman, but please raise your hand.
What I can really say is, you know, we look forward to collaboration because one can't be an expert in every field, but it's so important that we develop a better holistic understanding of disease. So I'm personally very interested in the systems immunology approach, where we take account of the genetics, the immunology, the molecular biology, and try and bring it all together. Because just looking at a cytokine or a cell in the lab, is never going to give you the full answers. So if we really want to be serious about tackling infections, and also scourges like long COVID, we have to have a very good understanding of the basic sciences behind it. And there we really welcome anybody who would like to collaborate. We're very open, especially in terms of the metabolomics. I think it's absolutely critical. I'm very excited to start linking the microbiome with the metabolomic work, because I think there will be some very interesting crosstalk. So please feel free to contact me. I'm going to be away until the 30th, so better not to contact me unless it's today. Rather contact me in July, please, because I'm going to leave my laptop at home and I'm going to go to Iceland for 10 days.
There is a question in the chat. Is the mucosal CD4 decline observed in non-progressors?
Yes, so that's a very, very interesting question. In humans, it's not been possible to do the same kind of biopsies, of course, than we can do in animals. So we don't know the extent. We know that there is some loss, but it doesn't seem to be as extensive. But a lot of this work is really based on proxy, not on direct biopsy work. You can imagine the ethics committee is a bit reluctant for us to, go into the mucosa unless it's really necessary for another clinical procedure. So the data that we do have out there may be a little bit skewed, but it seems as if they do also have an insult, but maybe not to the same extent as people who are fast progressors or normal progressors.
Okay. Are there any other questions before we let her go? So once again, thank you so much for joining us and sharing with us your story. It is amazing, and I hope everyone takes notes of how Prof Rossouw has approached things from multiple angles, how open she is to collaboration, to research. So I encourage you to reach out to her through an email. I do hope you enjoy your brief holiday in Iceland. We'll see you again in July. So you deserve a short rest.
Thank you. And thanks for this wonderful opportunity. It was really great to be able to share our work with you. So thank you.
Thank you. And please everyone join next week for the metabolomics aspect of Prof Rossouw's research. Crystal Roux, her student, will be presenting on that.